Okay, we're going to talk about ABO blood grouping. So in your notes, where it says ABO blood grouping, we're going to talk about that. Um, uh, your lab notes have some information about this ABO blood grouping there, and you're going to need to find that oh that little chart there. You can fill that out as you study, for the test and stuff like that, okay? So try to find that. Now, ABO blood grouping, we're going to try to find out the antigens on the surface of your red blood cells. The antigens are what are going to tell you what your blood type is. Tells you what your blood type is, okay? So that's what we're going to find out in here, and then we're going to find out that you have antibodies, which will be involved in that also, and that will be telling you something different. So here's your blood grouping. Blood typing is based on what antigens are on the surface of the red blood cell. All right, antigens are proteins on the surface of the red blood cell that are given to you by your parents. If you remember from general biology, you got the alleles for your blood type from your parents. It was multiple alleles. There's IA for red for type A, IB for type B blood, and little I for type O. But you only have two of those. There are three alleles, but you only have two. So it depends on what you're dealt, and that's going to give you the antigens on the surface of your red blood cell for whether your blood type is A, B, AB, or O. So there are three antigens we look at: antigen A, B, and the Rh antigen. We will look at the Rh antigen last. The antibody is the opposite letter, A or B, of the antigen. Since we have blood type A and blood type B, there's antibodies B and A, and is located in the plasma, so they're found in different areas. The antigens are on the surface of the red blood cell, and the antibodies are in the plasma of the blood. So in your lab handout, you're going to have something that's going to look somewhat like this. You have a little box, and that box has blood type A, B, AB, and O. Now, whatever your blood type is, that's what your antigen is. So you see that this person, let me get my little pointer out, is blood type A. So they have antigen A on the surface of their red blood cell. Now look over here to the left, and there's my red blood cell, and you see it has the little red triangles, which I have marked up here at the top. Little red triangles for antigen A. It has antigen A on there, so that's blood type A. We'll look at the little green pitchfork things in the in the plasma there, and those are antibody B. So antibody B. Now look at the top here, which says antigen on the surface of the red blood cell, antibody in the plasma. So you know exactly where they are. So whatever your blood type is, that's the antigen on the surface of your red blood cell. Your antibody is the opposite letter. Blood type B. Let's look at blood type B. They have antigen B on the surface of the red blood cell, which means they have antibody A in the plasma. Now over here, look at the red blood cell to the left. Here's the red blood cell, and there's the antigen B on the surface, the little green spheres, that's antigen B. And the little slingshots over here, red slingshots, those are antibody A. All right, let's look at blood type AB. AB means that there are both antigens, A and B, on the surface of the red blood cell. And you look at your cell on the left, and you see that, yeah, it's got both antigens A and B. But it has no antibodies. Now, the problem with that is that if you have the same letter antigen and antibody, it causes the cells to stick together. That's called agglutination. And that's little blood clots. It will block capillaries, so you don't want that to happen. So Blood type AB has both antigens and no antibodies, A or B. Blood type O. Blood type O has no antigens. O stands for 0. It has neither A nor B antigens. And since it has neither A nor B antigen on the surface of the red blood cell, it has both antibodies in the plasma. Both antibodies. So look to the left and you'll see there's a red blood cell. There are no antigens on the surface, but you have both antibodies in the plasma. Okay, so far we've looked at blood types that were negative. Now let's look at something before we go any further. In order to get your blood typing, we have these little bottles, and they're full of antibodies. Now, in lab, we would have had a slide that looks like this. It has 3 wells. We would have taken a stick with a little autolet. It would have punched your finger, and you would have put a drop of blood in each of these wells. You've gotten 3 toothpicks, 1 for each well, and I would have put in antibodies in each well. Now, all these droplets of blood are the same. They're identical. So we're going to test each one for something different. The first one here, you see anti-A, a drop of anti-A. Well, look what that is, antibodies. I'm putting antibodies in there to see if that drop of blood has antigen A in it. If it does, it's going to cause the cells to agglutinate or stick together. And you see on this slide, they did. So this person had antigen A in their blood. Well, look at the one below, one labeled B. This has, I'm going to put antibody B in there. So the yellow vial has antibody B, and we're seeing if there's antigen B in there. If there is, it's going to cause the cells to stick together. And you see that they did not stick together. So the antibody B did not find any antigen on the surface of the red blood cells. And so it just stayed like blood. But there's a third antigen that a person can have, and it's called the Rh antigen. You may or may not have it. This person has the Rh antigen on the surface of their red blood cells. This jar contains anti-D, which is anti-Rh positive antibodies. If you put the anti-Rh positive antibodies in this blood and it finds the Rh antigen, the antibody will bind to the antigen and cause agglutination. Now, usually agglutination in the Rh is a little bit finer than it is in the A and the B. Okay, so here's an example of that. Here's the blood type, and I had pushed blood type, but I've already shown you here. But you can tell that this blood type has been performed. There's no agglutination in A, so it's not A. There is agglutination in B. You can see the cells have clumped together. So they have antigen B. You put antibody B in there, it found antigen B and the cells agglutinated. You put anti-Rh positive antibody in here and nothing happened. So you look at the areas where you had agglutination and that tells you which antigens are present. In this case, the only one that agglutinated the blood was B. So it's B negative. The no Rh is negative. That's what that means. No Rh antigen. Now let's look at some basic blood types here, and then we're going to go back and look at the Rh. Here is somebody. Here's the blood being tested. Type AB contains antigens A and B. So they put antibody A in here. It found antigen A, agglutinated. They put antibody B in here. Found antigen B, it agglutinated. You read what agglutinates. A, B. So that person is blood type AB, like it says there. This next one, you see that nothing happened when antigen A was put in, but something happened when antibody A was put in, but something did happen when antibody B was put in. It found antigen B. So this one clumped, blood type B. Look at your third example. When antibody A was put in, it found antigen A and caused the cells to agglutinate. When antibody B was put in, it did not find antigen B, so nothing happened. You only read what clumps. A clumped is blood type A. In this last example, antibody A was put in this droplet, antibody B was put in this droplet of blood, and you see there's no agglutination in either one. So there's nothing to read. So that's blood type O. It doesn't have A or B. That stands for 0. It doesn't have A or B. Now let's look back up at the earlier presentation, part of this presentation, and look at something. And let's look at the first one, blood type A. Well, over here, these are the negatives. Because look, look over to the right, the top, these little blue spiky things represent the Rh antigen, the third antigen you can have. Well, look on the left. There are no Rh antigens here, so those are negative. But look on the right. This person has antigen A and the Rh antigens. So this person over here, on this side, on the right side, that's A positive, Rh positive, because it has that antigen. Let's look at the next one. Here's a B person. You look at the left-hand side, it just has the B antigen. That's B negative on the left. But on the right, it not only has the B antigen, but it's got the Rh antigen. That's B positive. Positive because it has the Rh antigen. This person's AB blood. Look on the left, it's AB negative. Just has antigens A and B on the surface. But the one on the right has antigens A, B, and the Rh. That's AB positive on the right-hand side. And the last one, blood type O, has no antigens, O negative on the left. But on the right-hand side, the only antigen on the surface is the Rh antigen, O positive. So let's look at some other little examples here. These were blood types that were made up, and you can tell what they are just by reading what agglutinates. A positive. See, there it is, A positive. B positive. Third one, A, B positive. Fourth one, ooh, nothing, that's O positive. At the bottom left, A negative. It's the only one that agglutinated. The second one over is B negative. It's the only one that agglutinated. The third one, AB negative. A and B agglutinated, but not the Rh negative. And the far right one, O negative. There was no agglutination in any of the wells. So it had none of the antigens, A, B, or Rh. Now, when you talk about universal donors and universal recipients, the universal donor is one that doesn't have any antibodies. That's going to be O negative. Because remember I told you, if blood has the same letter antigen and antibody, it'll cause agglutination. So if somebody needs some blood, you look at the person that's getting the blood, you look at their antibodies. And you don't want to give them any antigens that their antibodies can bind to. So if you give them O negative, there are no antigens. So O negative is the true universal donor. The true universal recipient is one that has all the antigens and no antibodies. So when you're looking at them, this AB positive, they've got all the antigens and they don't have any antibodies. So they're laying there and you say, okay, what antibodies do they have that would react with any antigens that we give them in blood? Well, they don't have any antibodies. So it doesn't matter what you give them. They've got all the antigens and no antibodies. So AB positive is the universal recipient. O negative is the true universal donor. See, I am O positive. I am not the true universal donor because I have a Rh antigen here. And I would be giving someone a foreign antigen if I gave them my blood and they were negative. So O negative is the true universal donor, AB positive, the true universal recipient. Now, as far as percentages in the environment, here's something that I found: blood population percentages and who each person can receive blood from or donate to. Well, I got this mainly for showing the blood types on the far left and the percentages in the population. You can see that all the negatives have the lowest numbers. About 15% of the population is negative. 15%. A majority of the population, like 85%, is Rh positive. So it kind of shows you who you can receive from. O can only receive from O, that's it. So O positive can take O positive or O negative. O negative can only take O negative. Remember, O negative is a true universal donor because it doesn't have any antibodies. So you'll see that all the bloods can take O negative. And AB can take any of them, especially AB positive. Blood transfusions. Let's look at this. A person with blood type A can a person with blood type A donate to a person with blood type B? First thing to look at is what antibodies does the recipient have? Blood type B, he's the one that's getting the blood. The type B blood recipient has antibody A. Remember, whatever your blood type is, whatever the antigen is, you've got the opposite letter antibody in your plasma. The red blood cells would agglutinate, so the answer is no. So if you have a person with blood type B there, they have antibody A that will bind to any antigen A that comes in. So if you give them blood type A, all the cells have antigen A, so they can't take that. It would cause agglutination, and many little clumps would flow through your bloodstream and block off capillaries. It could be extremely dangerous. Can a person with AB blood receive from a person with type O person? First thing, what antibodies does the recipient have? Talk about an AB person. The type O blood recipient has no antigens. The blood type AB has no antibodies. So yes. Okay. This would be O negative. It should be O negative. This would be AB positive. Then I reinstate it. O negative is the true universal donor because it has no antigens, A, B, or Rh, to bind to the recipient's antibodies. That's why it's O negative. It has none of the antigens that are necessary to bind on anything. Now we have AB positive. AB positive is the true universal recipient. The true universal recipient because it has no antibodies. It's got all the antigens, but no antibodies. So if you can give it any of the bloods because it has no antibodies to bind to any of the antigens which come in. Now it says also an Rh negative individual should never be given Rh positive blood. If they are, they just received a foreign antigen and they will build up anti-Rh positive antibodies. Oh, that's what it says. If that happens, the Rh negative person will build up anti-Rh positive antibodies. A second exposure to Rh positive blood could result in death due to the recipient's blood having massive agglutination. Now, let's look at this right here. When we talk about antigens, we're talking about a protein on the surface of the red blood cell. And an antibody is a protein in the plasma. And the Rh factor is an antigen on the surface of the red blood cell also. So there are three antigens, antigen A, B, and Rh. Rh positive means the antigen is present in that person's blood. Rh negative means that that antigen is not present in that person's blood. If an Rh negative individual is exposed to an Rh positive individual's blood, then he will produce anti-Rh positive antibodies. That's in your notes. Now what I'm showing you here is something called hemolytic disease of the newborn. Well, in just a minute I will. Here's the incompatibility problem here. Here's a positive father, a negative mother, and she's having a positive baby. Now she's carrying a positive baby. If there is a blood mixing across the placenta and the uterus, capillaries could mix the blood, the mother will build up anti-Rh positive antibodies. And that'll happen during the first birth. The first little guy gets out scot-free. But if it's the second one, the woman's blood, she will have anti-Rh positive antibodies that will attack the fetus. So let's look at this little next picture and we'll talk about this a little more. It says first pregnancy with Rh positive fetus and an Rh antibody treatment. What they do now to prevent the mother from producing the anti-Rh positive antibodies is to give her something called a RhoGAM shot. RHOGAM. That's your bottom blank there, called RhoGAM, and here it is on this picture, RHOGAM. So you can see number one, mother injected with RhoGAM immunoglobulin during pregnancy. So before birth, she gets a RhoGAM shot to block her ability to produce antibodies against a positive fetus. Look at number two, Rh positive red blood cells enter into the mother's circulation. Number three, anti-Rh antibodies bind and inactivate the fetal Rh antigens before they stimulate an immune response in the mother. After childbirth, she'll get another RhoGAM shot because oftentimes there's blood mixing, between the fetus, the baby that's being delivered, and the mother. So she'll get two RhoGAM shots, one prior to, delivery time, to make sure that she doesn't produce antibodies against a positive baby if she has one and if she's negative, and one after just in case the blood mixes to prevent her from producing those antibodies again. So hemolytic disease of the newborn, or erythroblastosis fetalis, that's what this is called, affects 1 out of 30 births, the result of an Rh negative mother having an Rh positive fetus. She must receive the anti-Rh gamma globulin antibody prep, also called RhoGAM, RHOGAM. Now this last slide, I kind of show you, put all the first 4 slides together to show the negative blood types on the left and the positive blood types on the right. The only difference between those two sides is the right side has a third antigen called the Rh antigen, which makes an individual positive, Rh positive.